It is my sincere pleasure to introduce Dr. Jeff Chamberlain. I know he has a great passion and dedication for understanding muscular dystrophies and developing therapies, specifically gene and cell therapies for muscular dystrophies, specifically currently Duchenne and LGMD2I. And the reason I know that is I worked for Jeff quite a few years ago as his postdoc, so it's my great pleasure to introduce him today. So Dr. Chamberlain is the McCaw Endowed Chair in Muscular Dystrophy and the Director of the Wellstone Muscular Dystrophy Research Center and the Professor of Neurology, Medicine, and Biochemistry at the University of Washington School of Medicine in Seattle. And today he's going to be talking about how progress with one muscular dystrophy advanced treatments for others. Dr. Chamberlain?
Thank you, Laura. It's a pleasure to be here today and to give you, share with you some of my thoughts on how research progress on muscle in general, as well as different therapies in particular, as are applied to different types of muscular dystrophy, can have a very wide benefit for a variety of different types of muscular dystrophy, even if those aren't the ones that were the initial target. So as you are all probably very aware of by now, there are thousands of human genetic or inherited diseases. There's at least 55 types of muscular dystrophy. More keep being identified. It's more than 30 types of limb girdle. And each one is, of course, caused by a mutation in a specific gene, so they all have slightly different or sometimes radically different effects on muscle function. But what we've learned from many of these often has a lot of crosstalk, and of course what we learn in basic studies of muscle biology is generally applicable to all the muscular dystrophies. And I'll give you a few examples of that. For example, most people know that the gene that causes Duchenne muscular dystrophy was the first muscular dystrophy gene that was identified back in 1985 by Lou Kunkel's lab. And this really allowed for the very first time detailed studies about how a single defective protein could have widespread and global effects on muscle function throughout the body. But more importantly for today, it's important to remember that the identification of the dystrophin protein led really very quickly to the identification of many other genes that were important in the development of the different muscular dystrophies, particularly a number of the subclasses of limb girdle muscular dystrophy. And one of the points I want to make today is to think a little bit about the tools that are being developed and used in basic muscle research, both to study what muscle tissue does, how it develops in a growing organism and a growing child, as well as how they are used to study the effects of the different muscular dystrophies. And of course, the most important of these is gene isolation and DNA sequencing, mutation detection, all of the DNA-based tools that we use in laboratories. These really started coming online, I guess really more in the late 70s, but really coming into play in the early 80s and the 90s. And this has led to the identification of well over 50 genes that are involved in different types of muscular dystrophy. Again, dystrophin was the first one identified in the late 80s. Other examples are the sarcoglycans that are responsible for several limb girdle muscular dystrophies were cloned in the 90s. And FKRP, responsible for limb girdle 2I, was cloned in the 2000s. Another really critical development was the ability to study muscle in a petri dish. The muscular dystrophies are a little hard to study in petri dishes because muscle is a very complex tissue. There's a lot of interactions with many other cell types, immunological cells, connective tissue cells, things like that. The ability, there's a lot of things that can be done in tissue culture, and it hasn't always been easy to study muscle in tissue culture. An important component of that is to understand the function of so-called adult muscle stem cells that are important for maintaining muscle in normal healthy muscle, as well as regenerating muscle after injury or in response to disease. And the first adult muscle stem cell was identified by Alex Morrow back in the 60s. But it was a number of years later before anyone figured out how to actively grow and maintain muscle cells in a petri dish And that was work done by my colleague Steve Hauschka in the early 1970s. Much later than that, the so-called pluripotent or induced pluripotent stem cells, really, which is a cell type that can be isolated from almost any cell in the body and converted into a muscle stem cell. That's work that came out in the 2000s. So it's taken a long time to really bring together all the different aspects of muscle biology in a petri dish. Other important aspects that are applicable to all muscular dystrophies are the ability to understand what are the thousands of different proteins that make up a muscle cell, how do those vary from one type of muscle to another, and how is that reflected in the RNA content in cells, and that's work that was originally pioneered by Eric Hoffman in the 90s and has continued to this day. Another aspect is really understanding how genes are turned on and off. A lot of muscle genes that make muscle proteins important for the muscular dystrophies are not used in any cell except for muscle. Others are used in every cell of the body, and this is all controlled by the process of these on-off switches, or the so-called muscle promoters. And again, this is work that's really been pioneered by Steve Hauschka, dating back from the early 80s to the present. What about animal models? You know, a lot of people ask me, why do you do anything in animals? Why not go right into patients? And one of the problems with that is, of course, most of the ideas that people have in laboratories of how to develop therapies don't ultimately pan out. And it's very helpful to have different systems that you can work through to try to figure out what are the bad ideas and what are the good ideas. And often that can be done by starting in tissue culture and then moving on to animal models, particularly mouse models. The original first animal model of a type of muscular dystrophy was the DY mouse, which is responsible for a form of congenital muscular dystrophy. And the second one, as far as I know, was the MDX mouse, which turned out to be a model for Duchenne muscular dystrophy. Viral vectors that are critical for gene therapy started coming online in the 90s, and they've gone through a lot of iterations, a lot of ups and downs, as we heard earlier today. There's a number of different viral vectors now. The so-called AAV vectors currently are the most useful ones for muscle, but there's others on the way that may be helpful too. And one of the more recent developments in terms of DNA editing, using the so-called CRISPR-Cas9 system, some of you may have heard of. DNA editing is something that was pioneered by people that really had no interest in muscle, but it's now being rapidly applied to muscle diseases and many other diseases. But there's a lot of challenges and problems with DNA editing that I'll talk about a little bit. And the people that are trying to solve these problems are working on many different systems, many different tissues, many different diseases, but this is all coming back to hopefully move this technology forward for the treatment of the muscular dystrophies. So another thing that it's important to emphasize is the links between the different muscular dystrophies. You know, most people are, of course, concerned with the dystrophies that are affecting their family members or friends and all that, but there are a lot of commonalities as well as significant differences. This is a bit of a complicated slide here, but the things I wanted to point out, if you can see that sort of purple band in the top middle of the slide, is a representation of the dystrophin protein and where it's localized within muscle cells. The so-called sarcolemma showing there that's in an arc going over the middle of the slide is the cell wall of a muscle cell. And again, dystrophin was the first protein found that caused a form of muscular dystrophy. And one of the first things that happened with that is groups such as Kevin Campbell and Eijiro Ozawa in Japan started doing a lot of studies of what does this dystrophin protein do? And what they found was that it binds to a number of other proteins. It links them and regulates their assembly and their proper localization within muscle cells. And one of the first things that was found is that dystrophin directly binds to 4 different proteins called the sarcoglycans, and it was soon found that each of those 4 proteins was responsible for a different type of limb girdle muscular dystrophy. The sarcoglycans and dystrophin both interact with a couple of proteins called the dystroglycans that are responsible for limb girdle muscular dystrophy 2P. They also interact with the laminins that are responsible for some congenital muscular dystrophies. And the list goes on and on. Dystroglycan, that is critical for the function of dystrophin, turns out to require more than 20 other proteins in order to properly assemble it and make it work right in muscle cells. And all 20 of those other proteins are each defective in a different form of limb girdle muscular dystrophy. So another aspect is really a lot of the research for many years, certainly still going on today, but the major form of muscle research that was going on before any of these genes were identified was studies of how does muscle develop in a developing organism, and how is it maintained in adulthood. And the understanding of this has been critical to trying to dissect what are the causes of the different muscular dystrophies, what are the things that are going wrong, and what is it you need to achieve to return that muscle to a normal, healthy self. This type of basic research, as I mentioned earlier, is what led to the isolation of muscle stem cells, and that has spurred tremendous interest over the last 30 years in trying to harness muscle stem cells as a way to repair muscle in different dystrophies. We still have a lot of progress that needs to be made in that area, but it's a very important approach. Studies of basic muscle function led directly to the development of gene therapy methods that we've been hearing about. And this started out just trying to understand how do muscle genes work? Well, how are they turned on and off? What is the structure of these muscle genes? And with that knowledge in hand, that created the tools necessary to start even thinking about gene therapy. And so, it's an often overlooked point, particularly as we're moving into clinical trials for many of these diseases, but I try to emphasize to people that continuing the basic research that has gotten to us to where we are today is critical to get to the next level, to the next generation of therapies, to perfect the therapies that maybe aren't all that working all that well yet. And I just cannot underemphasize or overemphasize the importance of basic muscle research. And one of the reasons I say that is that the funding opportunities to do basic research are rapidly declining, and it's something that we could certainly use a lot of help in trying to maintain research funding. The other important point when thinking about the different muscular dystrophies is that the proteins and the genes that create those proteins don't work in isolation, that nothing works by itself. All the proteins inside muscle, as well as really any other cell type, work in large teams. It's really a very collaborative environment inside of a muscle cell. And the links between these different proteins are typically referred to as pathways. And you can typically start off with protein A binds to protein B, which turns on protein C, and things like that. And a lot of the basic research in muscle biology over the years has gone into identifying these pathways, finding out which proteins interact with which other proteins. If you have a mutation of one protein, what effect does that have on other proteins? And this directly involves a lot of the limb girdle muscular dystrophies, because one of the ways that some of the genes have been identified over the years was by studying one disease and looking at one protein that was defective and realizing that there are several other proteins that weren't behaving properly either, and often it turned out that those other proteins were responsible for even a different type of muscular dystrophy. And again, maybe the best example of this is with dystrophin, which is defective in Duchenne muscular dystrophy. So the original study of Duchenne muscular dystrophy led to the identification of the dystrophin gene and then the dystrophin protein, and that led to the identification of a very large complex of proteins that all bind together that I was talking about just a minute ago. This is called the dystrophin complex of proteins. And in studying that complex of proteins led directly to the identification of so far 25 different genes that cause 25 different forms of limb girdle muscular dystrophy. So that all is a direct result of people that were studying Duchenne muscular dystrophy originally. There's also an equal, if not larger, number of proteins in muscle that cause other forms of limb girdle muscular dystrophy or congenital muscular dystrophy that are not directly related to the dystrophin protein, but those are coming out, and many of them have been cloned over the years. I've listed a few examples at the bottom of this slide, but there's, of course, many, many more. The other important thing to think about developing therapeutics is, as I was referring to earlier, the study of animal models. And the use of animals in research is, of course, a controversial area. It's becoming increasingly difficult to do. Many laboratories studying muscular dystrophy are getting increasingly harassed by different groups that are opposed to using animal research, and it's having a significant detrimental effect on the rate of progress. But it is important to keep in mind that animal models are the key to identifying new genes. They identify many of these pathways that I was referring to, and almost all initial testing of therapeutics happens in animal models. And lessons that we use, that we learn from the study of these different animal models has led directly to a number of important features in designing new therapies. I mean, it's developed a lot of the reagents that we use to study therapies, not only in the laboratory but in the clinic. Particularly a lot of immunological studies are really mainly focused on animal studies to try to predict what's going to happen in the clinic. It identifies many common features of disease, and a lot of the assays that are being used to monitor muscle strength and function were originally modeled and developed in these animal models. It's important to keep in mind, though, that no animal model completely mimics a human muscular dystrophy, so it's important to keep track of what you're seeing in a mouse, for example, versus what you're seeing in the clinic. And an important aspect of that is an observation that's not entirely clear why it is, but almost all mouse models of the muscular dystrophies have a milder disease than the human counterpart. And so it's something that you have to take into account when you're developing a therapy in a mouse. But there's also been a lot of work in trying to find ways to bring out the disease in the mice in order to make it better reflecting other dystrophies. And that's something that has a spin-off and makes it easier to study all the different muscular dystrophies. Most drug assays start out in the mouse, simple things like where does a drug go, how long does it last in the system. The Food and Drug Administration generally likes to see very thorough studies in at least one animal model before they'll approve a new therapy. And so you need that for proof of concept, safety studies, and eventual regulatory approval. So let me just talk briefly about a couple of different classes of therapies, because many of them don't have a lot in common, but they all have the common goal of trying to affect a modification or amelioration of a disease state. I often divide these into the classes of sort of conventional medicine, conventional drugs, small molecules, stem cell therapies, and the gene therapies. So let me go through each of those. Conventional drugs were the first line of defense. I'll just say right off the bat, there really hasn't been a great track record of pulling drugs off the shelf and having them being beneficial for the muscular dystrophies. Probably the most dramatic and the most useful drug that's ever come out of that are steroid hormones. It was work that was really pioneered by Jerry Mendell many years ago. But it is important to know how a drug can affect muscle, and that can lead to trying to address which drugs should be applied to which muscular dystrophies. Another example of a drug with mixed success are some of the drugs out there that are designed to modify muscle mass, like inhibitors of myostatin. These are things that can increase muscle mass. It's not clear they necessarily increase strength also. They were initially tested in Duchenne muscular dystrophy. Now they're in testing for a variety of other limb girdle muscular dystrophies. The results are not, in my opinion, particularly overwhelming, but they are interesting and potentially useful, particularly in conjunction with other therapies. There's a number of other classes of conventional drugs, if you will, that are being tested for different muscular dystrophies, things that will reduce how much growth of non-muscle tissue occurs in cells or fibrosis. I mentioned the muscle mass boosters, the steroid hormones. Tamoxifen is an interesting compound that's recently been proposed for testing in a couple of limb girdle muscular dystrophies. It's not entirely clear how it works, but it can lead to increases in muscle strength. And then there's a couple of emerging approaches of trying to modify how well a protein works. Particularly, there's a subclass of proteins known as enzymes. Typically, in the muscular dystrophies, let me back up a minute, you can think of proteins as playing two general roles. One is sort of a structural role, sort of like the 2x4s that hold up your house, and then another type of a protein is an enzyme that actually does chemical reactions and creates new things within a cell. With the enzymes, they're a particularly interesting class of proteins because they will take chemical A and convert it into chemical B, and that's important in one of these pathways that's essential for muscle growth or maintenance. And Key Lou's lab in North Carolina made a discovery a couple of years ago in studying the enzyme FKRP that's responsible for limb girdle muscular dystrophy 2I. And the substrate, or the starting chemical that enzyme works on, is called ribitol. And he showed that by increasing the amount of this ribitol present in muscles, that you can actually make a mutant protein work a little bit better, and that can lead to a milder version of the disease. That's the idea anyway. That's all work, again, that's been done in an animal model and a mouse model, and that's something that hopefully will be moving into clinical trials, and we'll see how well it works. If it does work, that is a concept that could then now be applied to at least 20 to 25 additional proteins that are defective in different types of muscular dystrophy. This whole idea of simply trying to get a defective protein to work a little bit better without any really kind of drug or medicine or genes, simply by increasing the concentration of a chemical that it uses to do its normal function inside a muscle cell. The favorite drug in a lot of modern research, particularly that going on in large pharmaceutical companies, is something people often refer to as small molecules. This is sort of a vague concept of synthetic compounds that you might make a library of millions of different chemical compounds that don't necessarily have any relationships to one another, and then putting them on muscle cells in petri dishes and ask, does the muscle cell get bigger, does it divide faster, or whatever assay you particularly want to look at. This is something that is usually tested in tissue culture using muscle stem cells, particularly these inducible or iPSC types of stem cells that can be converted to muscles in very large quantities. And that's an emerging and potentially very important platform for screening, although at the moment there hasn't been a lot that's come out of this type of platform and has shown success in the clinic, but I think that's something we're going to be hearing a lot more about in future years. Stem cell biology is something that has been pushed for at least 30 years. I remember attending a conference sponsored by the Muscular Dystrophy Association back in 1989, where it was thought that muscle stem cells were the key to curing all muscular dystrophies. So far that hasn't panned out, but there has been tremendous progress in the years since then in understanding what are muscle stem cells, how can you isolate them and grow them, and a problem has been getting enough of these stem cells to transplant them back into the body. We know that stem cell therapies can work. That's the basis of bone marrow transplants. You probably all know someone that has had leukemia or some type of cancer and they've had to undergo a bone marrow transplant. What that involves is introducing a blood stem cell back into a patient. And if we can do that with muscle, that could be tremendously beneficial because muscle stem cells have a unique ability to reproduce themselves and to merge back into diseased muscle and to repair that muscle. They can also bring in a new gene. You can have a form of gene therapy where you correct a patient's own stem cells and then transplant them back into that patient, or you can use donor stem cells from a close relative, things like that. A lot of these challenges are being solved, particularly the ability to get enough stem cells to do a transplant. We're getting there, but the major rate-limiting step in applying stem cells for therapies of the muscular dystrophies is that no one has figured out a way to get a stem cell to go all over the body and fix muscles body-wide. So far, they work really well if you do a single injection, and that's the way gene therapy started out too. We could only do a single injection and fix a very small amount of muscle. We've gone beyond that, obviously, but with stem cells, we still need to figure out a way to deliver them body-wide. The final form of therapy that I'm going to talk about is, of course, gene therapy. And this is an area that I've been working on myself for about 35 years. And initially, I must say, I like to point out that when I first started proposing the idea of using gene therapy for muscular dystrophy, a lot of people looked at me and told me I was completely insane and to give it up. I've heard that as recently as 5 years ago, but I think, as you've heard earlier, there's been tremendous progress with gene therapy. Hopefully it will continue to grow, become more useful and safer. The advantage of a gene therapy treatment is that it's designed to treat the cause of a disease. So many drugs and small molecules and chemicals that are out there are really designed to treat the symptoms of the disease. Gene therapy for a genetic disease where you've inherited a defective gene is really the only way to go in and actually treat the cause of the disease. The challenges to gene therapy are typically not unique to any particular form of muscular dystrophy. There's a lot of very common challenges. And as we solve these different challenges, it's going to make it easier to open up gene therapy approaches for all the different muscular dystrophies. So what are some of those challenges? Delivery. How do you deliver a gene to every muscle cell in the body? Almost 40% of the human body is muscle. That's a huge target area to deliver these new genes. How do you deliver a new gene to muscle without altering other tissues like the liver or the kidney? That's been a challenge. What about immunology? The biggest problem in the gene therapy field over the years has been the immune response, that the body often views this new gene coming in as a foreign invader, almost like a virus or a bacteria, and they can fight it off. That's been a big challenge, but one that is partly solved, not completely. Toxicity. If you deliver too much of a gene, sometimes it might function well at the beginning, but then it can cause some toxic problems because you're making too much of it, or maybe you're making not enough of it. How long is gene therapy going to last? Nobody knows the answer to that question. It depends a little bit on what tissue you're looking at. I think if you're looking at neurons, it's a very long-lived cell type. Some of the gene therapies targeting neurons may last many, many years, perhaps a lifetime. In muscle, I'm not as optimistic because muscle is a tissue that does turn over. Even normal, healthy muscle has a limited half-life. It undergoes regeneration processes with normal everyday life to maintain the muscle in response to exercise and things like that. Nobody knows how long the gene therapy is going to last. It's something that might need to be repeated, and there are a lot of challenges with doing a gene therapy more than once in an individual, but there's a lot of ongoing research that hopefully will solve those problems. Scale-up has been a big issue. Gene therapy is tremendously expensive. We've heard with the gene therapy for spinal muscular atrophy is being sold now for $2.1 million for one dose. Maybe you only need one dose. Hopefully not. Hopefully that's going to be the case. That's what it looks like so far, but that's a lot of money. Can we bring this down? Hopefully the efficiency of mass production as gene therapies is being applied to more and more diseases, these prices will come way down. And then there's a lot of ethical and regulatory challenges to gene therapy, some of which have arisen because of people overselling their research. Sometimes it's unclear how safe it is, but a lot of this comes from misunderstanding really what gene therapy is all about. And it's important, I think, for people in the field not only to advance the research, but to really try to convince people of the ethical approaches that are being followed. So there's a couple of different types of gene therapy. You've already heard about a lot of these. There's gene replacement therapy, which is the most commonly applied, where you're just trying to deliver a replacement gene for a defective gene in a disease. There's gene editing that's getting a lot of play these days in prominent scientific journals like the New York Times and the National Enquirer, places like that. Gene editing is designed to modify or fix a gene. It uses tools known as CRISPR-Cas9. There's a gene knockdown strategy, which is a little different than these others, which is designed to shut down a mutant gene. So some diseases are caused by genes that create a toxic product. And you cannot fix those diseases by bringing in a new gene. You've got to shut down the mutant gene. And there's a variety of ways to do that. And another approach is with exon skipping, which is designed to use different approaches to remove a part of a gene to make a mini gene. And by removing, if you remove the proper mutant part of the gene, you can often make a protein that is still functional. So there's been a lot of success, as we've heard about. There's been several gene therapies that have now been approved. They're available by prescription. This is a real breakthrough. That had never happened until 2 years ago. The initial gene therapies that got regulatory approval were involved with immunodeficiency and certain forms of inherited blindness, and with cancer, and more recently with hemophilia. Spinal muscular atrophy has been a tremendous success story, as we heard about earlier. Thanks to Jerry Mandel and Brian Kaspar and their tremendous work. That's now available via prescription. Cancer gene therapies using so-called CAR T-cell approaches. There's at least 2 of those that are available by prescription now, and there's emerging success with Duchenne muscular dystrophy as well, and hopefully this will carry on to many other types of muscular dystrophy. As I mentioned, gene therapy is designed to fix the cause of a disease. It has the potential for permanent treatment, although I wouldn't try to overplay that too much. It can be applied directly to muscle or to the genetic manipulation of muscle stem cells that are then transplanted into the body. And as I referred to a minute ago, it can be applied to both so-called dominantly and recessively inherited muscular dystrophies. I feel very strongly that developments in gene therapy for any muscular dystrophy are going to be directly applicable to all other muscular dystrophies, because so much of what we learn involves manufacturing, delivery, safety, things of this nature that are common common in all the muscular dystrophies. So what are some of these challenges that are being attacked for these different muscular dystrophies? I mentioned, how do you deliver a new gene to muscles all over the body? Well, as we've heard, AAV vectors are being widely used for that. These vectors were developed by taking a natural human virus, removing all the viral genes that make you sick, and replacing it with your therapeutic gene of interest. How do you make these delivery vehicles that have come from viruses go to muscle and not go to every single cell in the body? Well, sometimes you can't really do that, but you can at least control whether the virus is activated in muscle or another liver using these gene on-off switches that I was referring to each other. But which muscle do you need to target? Not all the muscular dystrophies affect the same muscles. Some affect almost every muscle in the body. Some only affect a subset of muscles. Some affect the heart. Some don't affect the heart. And regulating your gene therapy product and making sure it's working in the right cells and not causing problems in the wrong muscle cells can be a great challenge. It can affect the persistence of expression, the safety. Another thing to consider about is when you're dealing with genes, genes, of course, where the name comes from, genes form your genetic basis. That's what makes you a human, and that's what makes your children similar to you. But what you absolutely don't want to do, and what the regulatory agencies will not let you do, is to do a form of gene therapy that is going to cause that therapy to be passed on to future children that you may have. So it's critical that the therapy work in muscle and not in your germ cells, so that if something goes wrong, you're not affecting future generations in your family. So we've heard a lot about AAV, adeno-associated virus. So far, this is the preferred and really the only successful gene therapy vector for muscle diseases. It's responsible for most of the gene therapy approvals, but not all of them. There's many different types of AAV that are good for targeting muscle. There's even more types of AAV that are not very good for getting into muscle, so you have to choose your serotype carefully. They're relatively easy to produce. They're showing some promise for being able to be scaled up to enormous quantities. And it's the only gene delivery system that's been found that will target muscles body-wide. But their AAV is not a perfect system. There's some disadvantages. It's a very small delivery vehicle, so it won't hold large genes. That's what spurred a lot of the research that my lab has done for the last 30 years in trying to develop miniaturized versions of the dystrophin gene, and led to our discovery of what we call microdystrophins. AAV vectors don't work very well in stem cells because stem cells divide and they proliferate and they grow and grow, and AAV doesn't stick inside of a dividing cell. It's rapidly lost. Shown at the bottom of this slide is what an AAV vector looks like. There's only a teeny little piece shown in yellow on each side of the delivery vehicle that is from the original adeno-associated virus. The rest of it is a typical gene, a promoter, which is the on-off switch, the gene you're trying to deliver, and then a poly-A site, which we call a stop sign, because it basically tells the cell you've reached the end of the gene. Here's an example from work that we published in my laboratory. So this came from about 15 years ago, together with a brilliant postdoc in my lab named Paul Grigorovich. We made the initial discovery that AAV vectors could deliver genes body-wide and target muscles all over the body. Before that, people had only done single intramuscular injections of these AAV vectors, and we realized that there was a way to deliver genes body-wide with AAV. And that discovery, we originally did that in a mouse model for Duchenne muscular dystrophy, but this is now forming the basis for all gene therapies of all the different muscular dystrophies. So some of the remaining issues, though, again, we've heard a little bit of this earlier. What's the best AAV serotype to use? But what is the best on-off switch? I think this is a critical issue for different diseases. How strong of an on-off switch do you want? There are a lot of different types of muscle. There's heart muscle, there's limb muscles, there's smooth muscle. Even within your limb muscles, there's a lot of different subtypes of muscle. And these gene therapy vectors don't get into all of these different muscle types at the same efficiency, and none of the on-off switches that we have right now work equally well in all these different muscle types. So it's important to find the best ways to regulate how much of the protein you deliver to these different muscle cells. And the way that's emerged as the most promising so far is to use an on-off switch derived from a naturally occurring muscle gene known as the MCK gene, or the muscle creatine kinase gene. This is a gene that's only active in muscle. It doesn't work in dividing cells, it doesn't work in immune cells, it doesn't work in cancer cells. And all of the gene therapy switches that are being used in muscle gene therapy, or at least almost all of them, were developed by my colleague Steve Hauschka at the University of Washington. He's developed a whole bunch of different flavors of these switches together in our Wellstone Muscular Dystrophy Center. There's different ones called CK6, MHCK7, CK8, triple MCK. These have all come out of the Hauschka lab. They're now also being used not only for gene replacement, but they're being adapted for dominant diseases to try to create RNA molecules that will destroy a dominant gene, and they're now the favored on-off switch for the gene editing CRISPR-Cas9 studies that a lot of labs are doing. So the early way that Hauschka was developing these on-off switches was to try to find ones that were as strong as possible, to just blast out as much protein as possible, thinking delivery was going to be not very efficient. Well, it turns out the delivery has gotten a little more efficient than we thought it would be, and it turns out that it's equally possible that you make too much of your protein. So while you think you're about to treat this disease, you end up making so much of the normal gene product that it becomes toxic and it overwhelms the muscle cell. So you really got to be able to dial these genes on and off, up and down, to make the perfect amount of protein in the right muscle cells. And so Hauschka continues to make a lot of new versions of these on-off switches, and he's trying to adapt them for a variety of different limb girdle muscular dystrophies. So for example, there are some muscle diseases where you only want your gene to work in the heart. There are others where if your gene turns on in the heart, it's toxic and can cause heart disease. So he's developed some that only work in the heart, will only work in the limbs, will work in all of those. And I think these are going to be tremendously useful tools for gene therapy for a lot of diseases. So I'm going to close out a little bit by talking about gene editing, which uses the tools that is called CRISPR-Cas9. I usually try to avoid specific terms like that, but it's been in the newspaper so much, some of you may have heard about it. The idea behind CRISPR-Cas9 is this is a set of tools. It's not just one. It's a couple of things you need to deliver together that can bind. They can go into a cell and find the actual mutation that's causing a disease, home in on it, grab the mutant piece of DNA, and alter it. There's a lot of ways it can be altered. It can be just chopped out of your normal chromosomes. It can be replaced with a donor piece of DNA, and it can modify the piece of DNA in order to typically to bypass a mutation, to skip over it. There are methods where you can actually fix a mutant gene and return it to a completely normal state. Those don't work that well so far. They tend to not be very specific, but that's an ultimate goal, is that maybe you could completely fix a gene. But most of the CRISPR-Cas9 or gene editing strategies that you hear about these days are not really fixing a gene. They're simply making it more functional so that you can produce a little more of your protein to bypass the disease that you have. The efficiency of CRISPR-Cas9 or gene editing is not at the level of gene replacement yet, but hopefully it'll get there in the next few years. Here's an example of just a side-by-side comparison we did in my laboratory of comparing gene replacement for Duchenne muscular dystrophy with gene editing for Duchenne muscular dystrophy. And what I've shown here are some images of muscle where the green stain that you see is the dystrophin protein that is normally defective in Duchenne muscular dystrophy. When we deliver microdystrophin using an AAV vector from one of Hauschka's CK on-off switches, we can approach normal levels of dystrophin production in muscles throughout the body. When we do a similar method to affect gene editing of the dystrophin gene, we can produce some of the dystrophin protein, but not nearly as much with a simple gene replacement. So far, gene delivery is working better than gene replacement, than gene editing, but hopefully gene editing will catch up in the next couple of years. So what are some of the challenges of the gene editing field? I don't like to usually say this too much, but a lot of what you hear about gene editing is exaggerated, and don't believe everything you hear. There's a lot of people have personal interest for trying to push the technology, even if it isn't quite working as well as they think it is. One of the problems is there's a lot of off-target effects. Even if you can go in and fix a mutation in the gene that you're trying to target, generally you're modifying a few other genes at the same time. We're starting to get around that, but it's an issue. Immune responses are a big issue. It turns out that the tools that are necessary to affect gene editing, almost 80% of the human population has pre-existing immunity, and it could be difficult to deliver these things to those patients. The approaches that are being proposed to apply gene editing for the muscular dystrophies end up having the effect that the gene editing tools are going to exist in muscles for years and years. Now, the problem with that is that to fix a DNA mutation, you really only need to do it one time, and that can happen very quickly after the treatment. But these DNA modifying proteins that you're delivering stick around in the muscle for years and years and years, just like we hope that the gene replacement strategies will stick around for years and years. The problem with that is they're going to be sitting around doing things here and there to other genes that you don't necessarily want to modify, and nobody knows what the long-term consequences of that. So there's a lot of potential, well, there's a lot of tremendous advantages of gene editing, but what I would say is it's just not quite ready for prime time. So my last slide, I just want to talk about some of the future prospects for gene therapy for the limb girdle muscular dystrophies. Is basic science and the proof of principle have come a long way. We know a lot about what we need to do, but there's still work that needs to be done. The successes we're seeing so far in SMA, hemophilia, and DMD and things like that are giving us a lot of optimism. There are still issues that need to be resolved, but major efforts are now turning to manufacturing, scale-up, regulatory approval, and things like this that are going to be needed for all these different diseases. Gene therapy is moving into the clinic for a lot of different types of limb girdle muscular dystrophy now, but at the moment gene replacement is more advanced than gene editing, but we still need to make progress in gene editing. The other area is opposed to gene replacement is, of course, gene knockdown for the dominant diseases. That's lagging behind both of these other areas, but there's a lot of encouraging results coming out in the laboratory for dominant diseases, and hopefully we'll start seeing some human clinical trials for the dominant limb girdle muscular dystrophies in the future. And finally, you always want to keep in mind that no matter where we're at right now, there's certainly new ideas that could help the field. We need to keep our eyes open for new alternatives, not put all our eggs in one basket, and that's going to require continued basic research. So thank you very much for listening.